this drug, and therefore only those cells can survive. So that's a very simple trick, but it allows you to find, let's say, 1 or 2 cells in a million where this process really works. And that was really then the breakthrough in the field, so we did this, and this were the very first results. So you should imagine each of these little blue dots, this is a so-called resistant cell clone, and each of these cell clone contains an RNA of this mini genome, but on this plate were roughly 10 to the 7 cells, so 10 million cells. And what we got out were just 20 that survived. So a really power of selection. And when we then made these cell clones and expanded and made cell lines, our great delight, the virus was replicating extremely efficiently. And that, of course, was the starting point, because now for the first time we could study all the intracellular steps of the viral replication cycle, and we could then move on and study how the proteins are made, how the viral RNA is replicating, and I will show you just one example. This is a so-called HCV replication factory. This is a 3D reconstruction of a tomograph. So what you see here are membrane changes that are induced by viral infection. So what you see here in dark brown, this is the so-called endoplasmic reticulum, which is the endomembrane system of a cell. And all these light brown bubbles, these are replication bubbles that the virus is producing, and the replication then happens within these bubbles. So a pretty shielded compartment where the virus is replicating. However, more importantly, all these replicants contain pretty much all the primary drug targets. And since these replicants were so robust and easy to measure, you could simply add a drug, now let's say drug X, and you ask whether you hit the replicant. Very simple. And we then improved all these systems over many years, and we could adapt it to high throughput screens to allow the testing of millions of compounds in large high content screens. And it could also be adapted to rational drug design. So pretty much standard. And what finally came out of that were 2 enzymes in the virus that became the drug targets of all available antiviral therapy. The 3 drug targets are the NS3 protease, the NS5A replicase factor, and the NS5B polymerase. And there are 3 classes of antiviral drugs, of inhibitors. If you are interested in finding out what the drug target of these compounds is, you can simply look what's the name of these drugs. All the protease inhibitors are ending with previas. So we have tilaprevir, boceprevir, cimeprevir, so you know, okay, this is a protease inhibitor. We have all the NS5A targeting inhibitors. They are called the ASVIR, so we have daclatasvir, dipasvir, and so on. And we have polymerase inhibitors. These are the buvias, like sofosbuvir, which I think most people know. So then you directly know what the drug target is. So let's first take a look at the NS3 protease inhibitors. Now the protease inhibitors are targeting the HCV protease. And I don't want to go into all the biochemical details of that. I just want to mention one point, which in the early days of HCV research was a bit sobering. That was when the structure of the X-ray crystal structure of the protease was solved, it turned out that the substrate binding pocket is very flat. So what does that mean? It means the following. If you assume here this cliff is the surface of the protease, it's really difficult to stick something to it that really targets and blocks the enzyme. So like this cliffhanger here who tries to stick to the surface of the protease, but as soon as you blow a little bit, it falls off. So ideally you would like to have a target which is very hilly and has clefts and everything, so you can nicely dock in an inhibitor. But in fact the HCV protease was like that, so you try to dock something to it, but then the inhibitor is kind of dropping off simply because it doesn't have enough glue to stick to it. There was one interesting observation, and I only want to mention this as an anecdote, which is called the end product inhibition. What does it mean? So I already mentioned that the viral proteins are made from a long precursor, and the enzyme has to cleave that precursor into shorter pieces. Now the interesting point is when the enzyme has cleaved the protein, this part here of the cleavage reaction remains bound to the active site. And that's what is called end product inhibition. So you have here a precursor protein that is cleaved by the protease. And this short fragment here then remains bound very tightly to the active site of the protease. And what people then did was they used short peptides derived here from the product and made medicinal chemistry. So they introduced all sorts of chemical modification. And the first drug that came out from that was the protease inhibitor telaprevir. When you compare the structure of telaprevir with the natural substrate, you can see they look very, very similar. And this was actually the starting point of all the protease inhibitors based on this end product inhibition. That's why they are called peptidomimetics, because they look like a peptide, because they're derived from a peptide, but in fact they are not because of all the modifications. Now the downside of these inhibitors is telaprevir, they have a very high potential to rapidly select for resistance. And that's why second and third generation protease inhibitors have been developed. Cimeprevir, second generation, Ralf, which is one of the most potent protease inhibitor. This has a very high resistance barrier, so the virus has high difficulties to develop resistance against it, and it has a broad coverage of all HCV genotypes. In fact, Ralf still has a strong bias for genotype 1. That's why it's only recommended for treatment of patients with a genotype 1 infection. So the second class of inhibitor is targeting the NS5B polymerase. It's also called RDRP, RNA-dependent RNA polymerase. And there are two drug classes that have been developed. The first one, which I only want to mention because they are no longer in clinical use, are the non-nucleosidic inhibitor, and the only one that has ever been approved is dasabovir. So these drugs are binding to the enzyme, they block the enzyme, and very rapidly the virus develops resistance. So they have a low genetic barrier for resistance. That's why they are no longer used. That's why people shifted to the second class, which are the nucleosidic inhibitors, and that's essentially the class where sofosbuvir belongs to. And how do they work? Well, polymerase or nucleosidic inhibitors are often told to be polymerase inhibitors, but in fact they are not inhibitors, they're doing something different. This just comes when you take a closer look how they work. So this is now, let's assume, this is the viral RNA genome that needs to be amplified. And this polymerase is adding one nucleotide after the other until it adds a nucleoside analog like sofosbuvir. And this sofosbuvir is designed in a way that no further nucleotides can be added. So that's why they are causing a so-called chain termination. So in fact, they do not inhibit the enzyme as such, but you rather would like the enzyme to be active to allow the incorporation of sofosbuvir, but then the chain breaks off and the viral genome is no longer amplified. And that's how they are working. So the next nucleotide can no longer be added. There's a chain termination. That's like herpesvirus drugs as acyclovirs are working. The main point here is they have an extremely high barrier of resistance. In fact, clinically, there's almost no resistance against sofosbuvir, and that is simply because the nucleotides are incorporated into the active site of the enzyme, and to become resistant, the virus would have to mutate the active site, but when the active site is mutated, the enzyme is dead, and the virus is then also dead. So that's why they have an extremely high barrier for resistance. Now the last inhibitor class are the so-called ASVIRs, NS5A inhibitors, and I would say, personally, from all antiviral drugs that have ever been made, drug targets, these were really game changers because nobody expected to ever make drugs against NS5A. In fact, this protein was thought to be undruggable. So not possible to make drugs again simply because there is no enzymatic activity. So you have absolutely no clue what to inhibit because there's no enzyme. However, people did, and just to mention that, what NS5A is, it has a very complicated structure here, but the main point is this protein is doing many, many different things in the HCV replication cycle. It's binding RNA, it's required for building up these replication vesicles for assembly, it's blocking innate immunity, and so on. So this you can compare to a so-called Swiss Army knife. A Swiss Army knife usually is a knife where you have a knife, but it can do many things because it has a couple of tools within the same knife. So it's like NS5A, one knife, but it can do different jobs. But NS5A is really going to